Hello everyone, welcome to my channel. So today we are going to discuss further about the topic photosynthesis in higher plants. So photosynthesis is not just a one-line reaction, rather we can see there are total two reactions that is taking place, that is light reaction and dark reaction. And in this light reaction and dark reaction we will be seeing that there are multiple steps involved as well. So in today's class, we are going to focus on light reaction. But before that, let us know a difference between the light reaction and dark reaction. Light reaction, it is light dependent. So it needs light to occur. That's why it is taking place during the daytime. On the other hand, if we see the dark reaction, it is light independent. So if it is going to be light independent, it doesn't need light energy to power the reaction. That is why it can take place during the daytime as well as night time as well. So where is the location of this light reaction? We can see it is taking place in the thylakoids, that is the dish shaped structures that we have seen, right? So these are those dish shaped structures. And where is this dark reaction taking place? That is the stroma. Stroma basically means this is the chloroplast, let's say. So in the double membrane chloroplast, this whole area, the aqueous like semi fluid structure, right? We can see this is basically the stroma here. And in the stroma, we know thylakoids are stacked and it's present. So it is taking place in the stroma. And during the light reaction, we are going to see ATP and NADPH is going to be generated. Along with that, we are going to see it is going to also give us the oxygen by splitting of the water. And then in case of the dark reaction, we are going to see this NADPH and the ATP is going to be utilized, and it is going to help in conversion of the 6 molecules of the carbon dioxide to form one glucose molecule. So now students, we are going to know how the light reaction will be occurring in today's class. In the next class, we will be focusing on dark reaction. So in light reaction, as you can see, ATP is forming. We all know that the ATP that is forming by the process of phosphorylation. So what is phosphorylation? It is the process by which the ADP, that is adenosine diphosphate, it is given a phosphate group, and when it is given a phosphate group, it is creating ATP, that is adenosine triphosphate. This is phosphorylation, but you all can see here it is written photophosphorylation. So why this photo is added, word added? Because here we can see photosynthesis is happening. They are using the light energy, and using the light energy there will be synthesis of ATP. That is why the photon there, how we are getting the word photo, that is the light particle which is powering the entire process. That is why photophosphorylation, because ATP is synthesized with the help of the light. Now, this photophosphorylation takes place by two ways. First is our non-cyclic photophosphorylation, where we are going to see there is going to be electron transport, but it is going to be non-cyclic. And if you all remember, we have studied about the photosystem. So in non-cyclic, we will see photosystem 2 and photosystem 1 are going to participate. On the other hand, in the cyclic photophosphorylation, we are going to see that here there is going to be cyclic transport of the electron, and only PS1 is going to participate here. Now, like we all know that this photosystem, they are having pigments, reaction center, right? But how do they work, right? And how do pigments work after they are excited? Let's say there is a photosystem and there is a pigment also. Say, let's say pigment 1. So pigment 1 is having 500 nanometer of wavelength. So it is going to transfer the energy to which pigment? Is it going to be pigment 2 or pigment 3? Before we discuss about it, let us know that the pigments after excitation, they can give energy to the other pigment. So in which pigment it will give the energy? We know this relation, that the energy is inversely relationship with the wavelength. That means if the energy decreases, because we know when the pigment is going to be excited, so energy will decrease, then it is going to transfer the electron to the pigment of higher wavelength. Okay, so pigment gives energy to the pigment of higher wavelength. So it is going to give it to the higher wavelength. That means pigment 1 is going to give the energy to pigment 3 of 510 nanometer. So students, we know that PS2 is having 680 nanometer, right? And PS1 is having 700 nanometer. So during this non-cyclic transport, we will see PS2 is going to transport the electron go to the PS1. So PS2 to PS1, we will see electron transport. So let us now understand about the non-cyclic photophosphorylation. So we will see light of 600 nanometer or less than that is going to hit our, that is PS2. And we know PS2 is also called as 680, also P680. So in the PS2's reaction center, it is going to absorb the 680 nanometer light of wavelength, and we can see it is going to be excited. So once this is getting excited, we are going to see that there is going to be redox potential. Let me tell you. So redox potential is high here, and it is going to be lower as we go upwards. So we can see that there is going to be uphill movement. So as the electrons are going to be transported to the PEA. What is PEA? PEA basically primary electron acceptor, that is our pheophytin. Okay, so we can see that the P680 with the excited electron has a higher redox potential. So from high redox potential, it is going to the low redox potential. So uphill movement is happening, and we can notice it is going to lose an electron, and pheophytin is going to gain the electron, that is the primary electron acceptor. After this, it is going to pass to the plastoquinone, which is a mobile electron carrier, and after that it is going to pass through cytochrome B6F complex, then it is going to pass to plastocyanin, then it is going to pass to PS1. So PS1 we also know that, okay, let me reduce the, so we can see that PS1, we also know it is going to be excited by P700, right? So P680 is PS2 and PS1 is P700. So once the PS1 is getting excited, it is going to transfer the electron to the FES, that is our iron sulfur protein, and then it transferred to the soluble ferredoxin, that is FD. And now after transferring to the FD, we are going to see the final acceptor of the electron is our NADP plus. Okay, so NADP plus, using this electron and also using H plus, we are going to see it is forming NADP plus plus H plus. So we can see in the stroma region the FNR, that is the flavoprotein ferredoxin NADP reductase enzyme is present. Okay, so we can see here, so let us now come to this picture. So what we can notice, students, there is going to be a kind of uphill movement, and then we can see downhill movement, and then we can again see there is a uphill movement, right? So here, can't we see there is a Z scheme that is forming? We can say this is a Z scheme that has formed. Yes, so that is why this is called as also Z scheme as well. Okay, so this electron transport of non-cyclic electron transport is also called as Z scheme, where we see uphill, and here we can see there is going to be downhill, and again we are going to see uphill movement. All right, up till this it is clear. Now we are going to also see there is going to be a splitting of water. So you all must be wondering, ma'am, why the splitting of water is happening? Okay, so in the P680, that is in the PS2, we can see there is an oxygen evolving complex, OEC, oxygen evolving complex. So it is evolving oxygen. How it is happening? By the photolysis of the water. So the photolysis of water is happening in presence of the sunlight. So lysis means breaking down. Photo, in presence of the light, that is why photolysis of water, and it is yielding 4 protons or H plus ions and also 4 electrons and 1 molecule of oxygen by 2 molecules of water. Okay, so now out of this 4 electron that we have got, 2 of this are going to compensate the loss of the P680s, like 2 P680 photosystem, they have lost electrons, so it is going to be compensated by this 2 electron of the photolysis of water. So we can see oxygen is going to be evolved, that is why oxygen evolving complex. Now I would like to also tell you about the proton, how the lumen is becoming high H plus concentration, how the H plus concentration is becoming high in lumen. Firstly, you can all see that the H plus is released during the photolysis of water, and also we are going to see plastoquinone, which is reducing and taking the H plus, they are going to take the H plus from the stroma and it is going to transport it to the lumen region. This is one. Another thing is that if you see that NADPH is going to be from plus H plus, so it is going to take some H plus from the stroma region. So stroma region is going to have low H plus concentration. Okay. So students, in this non-cyclic transport, I hope all of you are clear that there is going to be a Z scheme, because P680 it is going to be excited, transfer the electron to the pheophytin, primary electron acceptor PEA, I am just writing, then plastoquinone, then cytochrome B6F complex, then plastocyanin, then finally to the PS1. After PS1 it is getting excited, it is going to transfer the electron to the iron sulfur protein, then to the ferridoxin, then finally to the NADP plus, where we can see it is going to be reduced by FNR into NADPH. But ATP is also forming. But for the ATP formation, you might see in your book it is written here that the ATP is forming. But for the ATP formation, there is ATP synthesis involved, and we are going to discuss about it. But first, let us discuss about the cyclic photophosphorylation. Here, as it suggests, cyclic movement of the electrons are happening during this ATP molecule formation. But when does it take place and where does it take place? So firstly, it takes place in the stroma lamellae. So if you all remember, this was the region, we tell it as the stroma lamellae, and in the stroma lamellae we can see only PS1 is present. There is no PS2. In the granathylakoids we can see, yes, the PS2, PS1 both are present. So non-cyclic photophosphorylations take place in the stroma lamellae, but it can also take place in the granathylakoids as well. When one light of the wavelength that is beyond 680, so beyond 680 we won't see that P680 or the PS2 will be excited. We will see PS1 will be excited that time. So it can take place in the granathylakoid, or let's say if there is no NADP available, we know NADP is the primary electron acceptor, final electron acceptor. So if it is not present, so we can see that PS1 is going to work, and electron flow by chance if it is stopped, okay, between the water and the NADP, then also we will see that the electron transport chain will occur, but it is going to be cyclic. So we see here that whole cycle is happening. The PS1 getting excited, transferring electron to the iron sulfur protein, then to the ferridoxin, then finally to the plastoquinone. It is not going to the NADP, okay, NADPH. Then we can see it is coming to cytochrome B6F complex, okay, and then it is coming to the plastocyanin, and then cycle back to our PS1 P700, okay. So now let us do a difference between these two. So we can notice, students, that cyclic and non-cyclic photophosphorylation is happening. So how it is happening? So the cyclic photophosphorylation, here we can see there is cyclic passage of the electron. In non-cyclic, it is going to be non-cyclic passage of electron. In case of the cyclic one, do we see any kind of splitting of water into oxygen? No. So it is an oxygenic photosynthesis. No oxygen is evolved. But in non-cyclic one, we will see oxygen is going to be evolved. And then we can see electron moves in a cyclic pattern, electron moves in non-cyclic pattern. PS1 is involved in the cyclic, and in PS2, both PS1 and PS2 both are involved. Then we can see the electron acceptor, that's the final one, is P700 in cyclic, and in case of our that is non-cyclic, it is NADP plus. So if you remember, during there is no NADP plus, then the non-cyclic photophosphorylation is not going to take place. So we will see cyclic will take place. Then photolysis of water doesn't occur as there is no splitting of the water. So here it is going to occur photolysis. We can see it undergoes under poor availability of carbon dioxide, but when the carbon dioxide is sufficient quantity, we will see that non-cyclic is going to take place. Synthesis of ATP occurs in both the cases, but NADPH is going to occur only during non-cyclic photophosphorylation, and it occurs in low intensity light, and we can see when the light intensity is optimum, non-cyclic photophosphorylation is taking place. Now the question comes, how the ATP is forming. So NADP, so we have understood. So ATP is forming due to ATP synthesis. So ATP synthesis has two parts. That is our CF1 and this is our CF0. So CF0 is embedded in the thylakoid membrane. This is our thylakoid membrane and it is forming a transmembrane channel that is going to carry out facilitate diffusion of the proton across the membrane. So the proton, if you all remembered, the lumen region and the stroma region, which was having deficiency of the proton or the H plus, we have seen that the stroma region was made deficient of the H plus. If you all remember, stroma low H plus content and lumen was having high H plus. So we will see that the protons that are going to be transported, they are going to be transported from the lumen to the stroma region, right? And for this we are going to see that our ATP synthesis like these channels are going to help us as well. And then we can see the CF1, it is going to protrude, you can see it is in the outer surface, right? And as it is in the outer surface of the thylakoid membrane, but how it is helping? So let's say when there is going to be breakdown of this proton gradient that is forming between lumen and the stroma region, it is going to cause a conformational change in the CF1 particle. As there is a change in the CF1 particle, we can see it is going to make the enzyme synthesis that is going to form several molecules of ATP. So not only there is going to be transport, that is facilitated diffusion of the H plus. So H pluses are going to come here because we know here in the stroma there is low H plus concentration and in the lumen we have the high H plus concentration. But on the top of that, ATP is going to be released as well. So we can see it allows diffusion of the proton one across the membrane and then it releases energy, and due to this energy we are going to see it is going to activate ATP synthesis and it is going to form ATP. But this ATP synthesis forming by the chemiosmosis, or we can say chemiosmotic hypothesis, was also introduced or proposed by Peter Michel. For chemiosmosis there are total four things that are required. What are those? That is firstly, so just a second class, I'm going to shift the screen. Yes, so now it's fine. Okay, so we can see here that for this there are four things important. Membrane, yes, we can see here we have our thylakoid membrane that is being utilized here. Okay, then we have our proton pump. Yes, we can see proton pumping is happening, and proton gradient. Yes, proton gradient, like we can see stroma low H plus content, and we can see that the lumen region is having high H plus content, right? We can notice this. And ATP synthesis, yes, ATP synthesis also there. We can see this is there as well. So I hope it is now clear that all these four things are present in our ATP synthesis mechanism. Now the proton gradient, if you all recall now, I was telling that there was proton gradient created. So stroma was having low H plus because all the H plus that was there in the stroma, let's say the plastoquinone, it is going to take it from the stroma region and it is going to transport it to the lumen. This is one. So plastoquinone releases the proton to the lumen side, and also so when the splitting of water is taking place, H plus is also released. So hydrogen ion produced during splitting of water accumulate in the region of the lumen. And also if you see the NADP plus, which is going to accept the electron, it is going to be reduced as well. So the protons are also removed from the stroma because in the stroma region the H plus which were there, they are going to be utilized. As the H plus are utilized, so there will be low H plus. So that's why we can see reduction of the NADP to NADPH plus H plus is going to also reduce the proton in the stroma region, and that is why we can see this gradient is forming. And how this gradient is broken? The gradient is broken down due to the movement of this proton, as the protons are moving, we will see there is going to be a proton motive force which is going to release the ATP, adenosine triphosphate. So as it is moving across the membrane to the stroma, we can see transmembrane channel of the CF0 of the ATP synthesis is going to help in production of the ATP. I hope after this it is clear to all of you. Okay, so in today's class, we have understood how the photophosphorylation is happening in case of the ATP formation, and also how NADPH is going to be produced as well. Then how the ATP is forming, it is like taking the help of the proton gradient that is forming. So chemiosmotic hypothesis is going to help us in the ATP synthesis formation. Okay, so students, have a nice day everyone. Take care and revise. And next class, we are going to discuss about how the ATP and the NADP that is forming, we are going to utilize them. Okay, so thank you everyone for attending today's class. Bye.